You can start. Okay, intelligent design against naturalistic evolution. What is the better explanation of origins? There are 4 facts which all agree upon in regards of evolution, and there are 2 which are disputed between the proponents of intelligent design and the proponents of evolution, namely common ancestry and the origin of biodiversity and higher organismal form. In regards of common ancestry, there are 6 major points which evidence that the claim is not true. The major transitions in evolution are divided in 6 major steps. In the evolutionary narrative, at the root of the evolutionary tree, there is the split of the 3 domains of life. The origin of eukaryotic cells is acknowledged to be a major evolutionary problem. Eukaryotic cells function on different physical principles compared to prokaryotic cells, which is directly due to their size, which is up to 1,000 times larger in diameter and having a volume size up to 15,000 times larger. The diversity of the outcomes of phylogenetic analysis has led to considerable frustration and phylogenomic impasse owing to the inadequacy of the available phylogenetic methods to remove the ambiguity of relationships. Still today, most biology textbooks tell us that organelles such as chloroplasts and mitochondria evolved through endosymbiosis, a hypothesis promoted by Lynn Margulis, and that is supposed to explain the origin of eukaryotes. Because mitochondria look like prokaryotes, it was assumed that eukaryotic cells came into existence when one prokaryotic swallowed another prokaryote. A problem with this idea is that prokaryotic cells lack the ability to swallow other cells. Eukaryotic cells can, but every eukaryote that we know has mitochondria. It's not clear which came first, the ability to swallow other cells or the mitochondria. Then in 2015, some science papers announced that the link between prokaryotes and eukaryotes had been found. And then in Nature magazine, they published that modern phylogenetics indicates that the host, which engulfed another bacteria to become mitochondria, was an archaeon. Only 1 year later, they had to acknowledge that there was no relatedness and that the results indicated that Lokiarchaea did not correspond to Eukarya. There is a considerable number of innovations that would have had to take place to transition and evolve from prokaryotic to eukaryotic cells. I am listing over 40. One of the major problems is the origin of organelles in eukaryotic cells. The Golgi apparatus, the nucleus, the mitochondrion, etc., require information in the organelle from the mother cell to make organelles for the next generation, its daughter cells. That raises the question how these organelles evolved if the first could not inherit the information from the mother cell. Daughter cell membranes come only from mother cell membranes. When a cell reproduces by division, it has to duplicate its organelles in addition to its chromosomes. In general, cells do this by incorporating new molecules into the existing organelles, thereby enlarging them, and the enlarged organelles then divide and are distributed to the daughter cells. Thus, each daughter cell inherits a complete set of specialized cell membranes from its mother. During the process, the organelles are enlarged and subsequently must divide. But how does this happen? We all know how a boa kills its prey, by constriction. Amazingly, there is a set of proteins who perform their task like a boa constriction. They are called dynamine and operate by an amazing process called organelle constriction. They possess the remarkable property of assembling into contractile helical polymers that wrap around the neck of an organelle. They perform the challenging job of constricting the organelles until they are separated. This is just one example of many highly orchestrated and finely adjusted processes that have to take place during organelle constriction. In my understanding, this process had to emerge fully functional right from the beginning. A stepwise evolutionary process seems not feasible. To me, this looks like amazing molecular engineering and design. Another huge problem is the origin of mitotic cell division, which is one of the most fundamental tasks of eukaryotic cells. Cells have intricate and tightly regulated machinery to ensure that mitosis occurs with appropriate frequency and high fidelity. At least 14 cell cycle regulators are essential. If one of those regulators is missing, the cell cycle cannot be completed. Therefore, the regulation of the cell cycle is irreducibly complex. To my next point, 5 geochemical energy cycles are essential for advanced life forms on Earth. That creates a huge problem for evolutionary scenarios. How did these cycles get off the hook? These are gigantic interdependent systems where if one part of the cycle is missing, nothing goes. And they are also interdependent between each other. The 3 main cycles are the water cycle, the carbon cycle, and the nitrogen cycle. These 3 cycles, working in balance, are responsible replenishing the ecosystem with the nutrients necessary to sustain life. If any of these 3 cycles should become unbalanced, the effects on the ecosystem are catastrophic. Without cyanobacteria, not enough fixed nitrogen would be available. Without fixed nitrogen, no DNA, no amino acids, no proteins can be synthesized, and no life would probably exist. This is an interdependent system. It cannot have evolved in small steps. All must exist at once. In the evolutionary narrative, life started in an atmosphere that was anoxic, and only anaerobic life forms existed. It is supposed that life started about 4 billion years ago, and about 2.5 billion years ago, oxygen started to fill the atmosphere through cyanobacteria, performing photosynthesis, and by this, they produced their food, glucose. Oxygenic photosynthesis and nitrogen fixation are conflicting metabolic systems. Nitrogenase is the enzyme that fixes nitrogen, but is oxygen sensitive. As soon as it is exposed to oxygen, the enzymatic reaction shuts down, and oxygen produces as well hydroxyl radicals which wrecks DNA. So as soon as cyanobacteria would have started to produce oxygen, they would have killed themselves. So why would cyanobacteria have evolved protective enzymes if the need for them didn't even exist yet? Nick Lane describes the dilemma in the book Oxygen, the Molecule That Made the World. Before cells could commit to oxygenic photosynthesis, they must have learned to deal with its toxic waste, or they would surely have been killed. But how could they adapt to oxygen if they were not yet producing it? The origin of oxygenic photosynthesis very early in the evolutionary timeline is another huge unsolved problem. There would be no advanced life forms on Earth without this pathway, which is almost the most complex biosynthesis process known. In photosynthesis, 26 protein complexes and enzymes are required. A good part of the protein complexes is uniquely used in photosynthesis. The pathway must go all the way through, and all steps are required. Otherwise, glucose, which is what cyanobacteria consume for food, is not produced. The supposed evolution of multicellular life forms from simpler unicellular microbes is another pivotal moment in the history of biology on Earth. Biologists can provide plenty of guesses about how multicellular organisms could have evolved from unicellular organisms. Today, most plants, animals, and fungi are composed of multiple cells that work together as a single being. The fact is that science is basically in the dark of how this transition actually happened. And there is a good reason for this. Explaining organismal architecture depends on explaining how organs, tissues, and cells form and gain shape. On the lowest level, there are at least 19 different specifications required for each single cell. So now to the last and most important question. If unguided evolutionary mechanisms do not explain the origin of organismal form, what does? The real mechanisms to explain organismal complexity are prescriptive, pre-programmed, instructional, complex information encoded through various genetic codes and about 30 epigenetic codes, signaling and communication using various signaling networks that act on a systems level and are all involved in organismal development, responding to environmental cues and nutrition demands. Here, I am listing 19 different mechanisms which operate on a systems level. That brings us, without any doubt, to an intelligent designer. Thank you.
Awesome. Just on time there.